Hey everyone, welcome to the next Lanterna IB Biology revision video. Today we're going to look not at the cell on a level of organelles and the processes going on, but we're going to zoom in and we're going to talk about molecular biology. So in the introduction video, we already had a little intro to this, and we talked about atoms, molecules, what all of that really means. So if you haven't already, please go and watch the introduction video to this revision series because it explains the most important concepts sort of as a basis. So what is an atom, what are molecules, etc. And then we're going to jump right in. So when we talk about molecular biology, what we're describing are metabolic pathways. So metabolism, which we also discussed in video 1, is one of the functions of life. What metabolism means is essentially all the chemical processes that are going on in an organism at any particular point. And these metabolic pathways, these functions of metabolism, can be anabolic or catabolic. Anabolic pathways or anabolism means that we are building complex large molecules from simple molecules. So we're putting energy into creating these complex molecules. And the kind of reaction we need for this is called a condensation reaction, because we're joining organic molecules together and we're actually creating water. So a condensation reaction means we are joining molecules together and water is created as sort of a waste product. On the other hand, we have catabolism or catabolic pathways. Those are the ones where we break down molecules. So you can think of that as digestion and what happens to the molecules that you ingest later on. So that means that these molecules will be broken down and we actually release energy that can then be used elsewhere in the cell and the organism. And the reaction we mostly use for this is hydrolysis. And this is the opposite of a condensation reaction. So we actually take a water molecule, H2O, and we use it to split apart molecules into simpler, smaller molecules. Now, when we look at these molecules that are central to metabolism and central to all living organisms, we can't really get around the element carbon, because carbon atoms are central to all of these compounds, and they are called organic compounds. So what's an organic compound? It is a molecule, a larger molecule, where carbon is the central element. But careful, just because a molecule has carbon in it doesn't automatically mean that it is an organic compound. For instance, CO2, so atmospheric carbon dioxide, is definitely found in living organisms, but it is not an organic compound. What also makes an organic compound is that there are covalent bonds between carbon and other elements, for instance, hydrogen, oxygen, or nitrogen. So what is a covalent bond then? A covalent bond is when two atoms form a connection or a bond based on the sharing of electrons. And carbon can form 4 covalent bonds. And that's why it is so central to forming large and complex molecules, because we can easily create very strong and long-lasting bonds between, for instance, oxygen, hydrogen, and these very central carbon molecules. Many of those molecules that we're going to look at are actually made up of very easy building blocks. And these building blocks we call monomers. And if we put a lot of monomers together, we get what we call a polymer. So mono, one, poly, many. That means we are creating out of the many building blocks a long, often chain molecule or a different structured, complex, large molecule. That is going on a lot, right? We talked about catabolic and anabolic pathways. Those are the pathways that are important for either creating a large molecule or breaking one of those large polymers down into monomers again. So what are our examples? What are we going to look at in the rest of the video? We are going to look at carbohydrates. So pasta, an apple, table sugar, those are mostly carbohydrates. Then we're going to look at proteins. So a steak, or if you prefer the vegetarian alternative, chickpeas. And then we're also going to look at lipids, so that would be butter or coconut oil. And then finally also nucleic acids, which people often forget, but they form part of these 4 central categories of organic compounds. Our first category are going to be the carbohydrates. So carbohydrates are sort of the quintessential organic compound. They're just made up of carbon, hydrogen, and oxygen. If we link them together in certain variations, they form ring structures. So if you look at sugars, those are usually ring structures like the glucose molecule over here. A glucose molecule like this is a monomer of carbohydrates. And specifically here we call them monosaccharides, so simple sugars. Other examples include fructose or galactose. From the name OSE at the end, you can already see, again, it's very simple in biology. Once you get the hang, you can recognize molecules just by their ending. Now, if we link two of those monosaccharides together, for instance, this glucose, which is a hexose, so a 6-carbon sugar. We could also take a pentose, a 5-carbon sugar, like the one that is present in DNA, which we'll look at later. If we take two monosaccharides and we join them together, again, that is a condensation reaction, and water is created in the process. Then we have a disaccharide. So di meaning two, so it's just a 2-sugar compound. And an example of this would be sucrose, which is a disaccharide made up of two monosaccharides, which are glucose and fructose. But of course, we can also link more of these monomers together, not just forming a disaccharide, but actually forming very, very long chains. And once we do that, we get to what's called complex carbohydrates. So now we're not talking about stuff like the fruit sugar in an apple or the table sugar. We're talking about pasta, for instance, or the starch found in potatoes. So what are the three categories that are important here, or the three types of complex carbohydrates? Just like I said, it's starch, for instance. Starch is how plants store these monosaccharides so that the cells of the plant can use them later. The plant also stores monosaccharides or carbohydrates in general in another way, namely in cellulose, which we have talked about because it forms the plant cell wall. So we have cellulose and starch in the plant cell. In animal cells, carbohydrates are stored into complex forms in what's called glycogen. That usually happens in the liver, and that'll be important again later once we talk about hormones in the body that regulate how much sugar is released or stored in there. Now we're coming to the next group, and that group is called proteins. So proteins, you probably also know from your everyday life, because people who go to the gym a lot always talk about eating proteins, and then they eat really disgusting egg white omelets or something. But proteins come in a variety of forms. So for instance, if you touch your hair, that is made up of proteins. Keratin, for example, which gives the hair its structure. Proteins are also very, very important because a lot of hormones are protein-based, and proteins have a very, very central role everywhere because enzymes are proteins. That'll be something that we'll get into later more. Now let's just look at what actually makes up a protein. If we say that proteins are polymers, what are the monomers? The monomer of a protein, which we can also call a polypeptide, is an amino acid. So an amino acid is what we have over here, sort of that structure. And what we can see here is that there is an N in there. The N stands for the element nitrogen. That group, the NH2, is called an amine group. That's what gives the amino acid its name. Then on the other side, we have a COOH group, which is called a carboxylic acid or carboxyl group. And that is the acid part of amino acid. Then we have a hydrogen atom down there, and finally a rest group, that's the R. The R doesn't stand for any particular atom here, but for a side chain or rest group, because there are actually many different amino acids, and whatever is in that side or rest group determines which amino acid we're looking at. But in terms of the amine group and the carboxylic acid group, they're all the same. And of course, we are going to join these monomers together. And once we do that, if we take two amino acids, we are going to have a condensation reaction, so forming a covalent bond between two molecules. And in the process of doing that, we are creating water, so H2O. And when we're doing this, we are connecting the amine group of one amino acid with the carboxyl or the carboxylic acid group of another amino acid. And that bond is called a peptide bond. And so those two amino acids together, we're calling a dipeptide. Now, if we do this again and again and again and again, now we have a polypeptide. So again, we're going from one monomer, combining it with another monomer. Now we have a dipeptide. And then at some point we have the polymer, which here is called a polypeptide. But we're actually not going to stop there, because protein structure is something very complicated. But we need to understand it in order to understand what proteins can do, for instance, when they are enzymes. If we have a long polypeptide chain, that is what we call the primary structure of a protein. But as primary sort of first structure tells you, we can get to a secondary structure. These secondary structures actually formed because hydrogen bonds are formed between the carboxyl groups and the amine groups of the amino acid that are actually not adjacent in the polypeptide. But if we have a very long chain and it folds, they can of course also interact. And that's why we have so-called beta-pleated sheets and alpha helices as the most common type of secondary protein structure. And we get a very complex 3D structure when we get to the tertiary structure, which then includes interactions between our rest groups or side chains of the amino acids. And there we have lots of different bondings. We have disulfide bridges, we have ionic bonds, covalent bonding, hydrogen bonding, everything that you can imagine in terms of weird chemistry words that probably don't mean anything to you because you chose to have biology, not chemistry. But what's important here is very strong bonds and a lot of different possible configurations because all of these side chains are different in the different amino acids. And now the tertiary structure is actually the most complex structure that one polypeptide can get to. But we have a quaternary structure in some complex proteins, and that is when we combine multiple tertiary structures together, so multiple polypeptide chains. And that can even be combined with what's called a prosthetic group. So that is a non-polypeptide molecule that combines with our polypeptide chains. The most important example of this, which you'll hear a lot, is hemoglobin or hemoglobin, whatever pronunciation you prefer. That is a molecule which is a protein, but it is a very complex protein because it is in a quaternary structure. It consists of 4 polypeptide chains and a heme or heme group, which is a prosthetic group, so a non-polypeptide. And hemoglobin is responsible for carrying oxygen, so O2, in the blood. Again, 2 things to remember about proteins that will be very important later on during the course and during other videos. Proteins are made up of amino acids and the sequence of amino acids. So how we combine amino acids to form a polypeptide, that is coded for by genes. So last week, when we talked about mitosis, we talked about genetic material, DNA, that is coiled and then represented in chromosomes. And so on a chromosome, we have the different genes, right? Don't worry if you don't know this already, we'll get to that later. But these genes and the information stored sort of on the DNA, that is what codes for the sequence of amino acids in a protein, in a polypeptide chain. That connection is very important to realize early on, and I'll explain it in much more detail later when we get there. So our next group of organic compounds is lipids, and for these it's a bit more complicated to describe what they're made up of because they don't have very clear monomers, not like for carbohydrates where we have the monosaccharides or for proteins where we have the amino acids. For lipids it's a bit more tricky, but what we do have are fatty acids, and these fatty acids form part of many of the more simple lipids, for instance triglycerides, which we mentioned before. In triglycerides, we have a glycerol group and 3 fatty acids. So let's start with these fatty acids. What are they? Fatty acids are just very long hydrocarbon chains, so chains where we have a carbon backbone and then many hydrogen atoms and no oxygen here, right? The oxygen is only found at the very beginning of the fatty acid, as we can see in this representation here. We make a very important differentiation between fatty acids, between saturated and unsaturated fatty acids. And you know this because you've probably heard it somewhere if you're a bit into dietary requirements and such, because there's health differences as to whether you consume a lot of unsaturated or saturated fatty acids. The most important thing to remember in terms of recognizing them and knowing about their molecular structure is that saturated fatty acids have no double bonds between the carbons in the carbon chain. However, unsaturated fatty acids are not saturated with a lot of hydrogens. They might have double bonds between the carbon atoms. So as you can see over here, that's the most important and crucial difference here. The saturated fatty acids are usually solid at room temperature because they're not as flexible, they can't bend. And so that is something like butter or any fat that comes from animal sources, really. However, if you have unsaturated fatty acids, that will be something like olive oil or any other plant oil, except for instance, coconut oil, which has a lot of saturated fatty acids, which is why at room temperature, it's more of a solid. Maybe also interestingly, you might ask, well, why do we have lipids and why do we have carbohydrates? You know, proteins, they're very different, but those two seem similar. And yes, they are similar and we can actually use both for energy storage, but it's a very different kind of energy storage, as you probably also know from what people say about eating carbs versus eating fat or lipids. So some key points here to remember are that you can store energy more easily or for the long term in lipids. However, if you need energy for the short term, you're going to look at carbohydrates. Also, carbohydrates can be soluble in water, whereas lipids, as you probably know because they're fats, they're fatty acids, are not soluble in water. So these are just a few differences, but differences that are very crucial for molecular biology. And now for the fourth and last category of organic compounds, we are going to look at nucleic acids. So nucleic acids are DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. DNA, as we've mentioned last week and before, is the genetic material in most organisms. And so it's very central, obviously. However, RNA plays a crucial role as well because RNA is responsible for actually translating from the genetic material to then forming a polypeptide chain from what's stored in the genetic material. Again, that is something to be discussed later, but we will definitely get there when we talk about genetics and such. So what are the monomers of these very complex molecules, the nucleic acids? We call them nucleotides. And the nucleotides have a very similar structure for both DNA and RNA. There are just a few differences. Let's look at DNA first. In DNA, we have a 5-carbon sugar, so a pentose, which is called deoxyribose. Then we have a phosphate group, so PO4. And then finally, we have a nitrogenous base. So a base, and again, if you're not into chemistry, a base is sort of the opposite of an acid, right? And a nitrogenous base, because there's nitrogen in there, and there are 4 different types for DNA: adenine, thymine, cytosine, and guanine. For RNA, this is a bit different because we have a different sugar, essentially. It's also a 5-carbon sugar, or a pentose. But here, it is not deoxyribose, it is just ribose. So there's one more oxygen in the molecular structure. Then we also have the phosphate group, and we have nitrogenous bases. However, they are different ones here because we substitute one of them. We have adenine, guanine, and cytosine, but there is no thymine. Instead, we have uracil. So also 4, but no thymine. Instead, we have uracil in RNA. Another crucial difference here is that DNA once we combine our monomers, our nucleotides together, will form a double strand. So the first thing that happens is that we have so-called phosphodiester bonds between the sugars and the phosphate groups. And that forms a chain, essentially. But for DNA, what also happens is that we have hydrogen bonds between the nitrogenous bases of one of these strands with opposing nitrogenous bases on another strand. So we have a ladder, and that ladder is actually wound, so we have what's called a double helix structure. For RNA, we just have a single strand in most cases, and that is one of the main differences here. Also what they're used for, as we've mentioned before. Don't worry if this seems a bit much at this point. There's a lot to learn about DNA, so what nitrogenous bases go together and why that is important, how that works, we'll get to that in another video.